My name is Jennifer Steeden, and I'm going to talk about some of the ways in which we can translate novel reconstruction technologies into the clinical environment. So magnetic resonance imaging is known to be an extremely valuable imaging technique and has become the gold standard measurement for diagnosis and management of many diseases across the body. However, it's a relatively slow imaging technique which results in long scan times and makes it sensitive to motion. We are often able to achieve good quality imaging through patient cooperation, including breath holding and the use of ECG gating, in this case of cardiac imaging. However, there are a large number of sick patients where the reality is the images are compromised by motion, as in the case on the right. Therefore, there's a great interest in speeding up MRI. There are two main ways in which we can speed up MRI imaging. The first is to use non-Cartesian trajectories, which collect K-space in a more efficient way, including echoplanar imaging, spirals and radial readouts as shown here. Of course, the other way is to miss out some of the data points, undersampling. However, this violates the Nyquist limit, which results in artifacts in the images which are dependent on the sampling pattern, as you can see here. We need to remove these artifacts in the reconstruction to recover clinically useful images. So I promise this is my only slide with equations. But remember, in the basic MR experiment, the data we collect, B, is equal to the object we're imaging, M, multiplied by the encoding operator, E, which includes things like the core sensitivity profiles, the Fourier transform, and the sampling mask. Therefore, the image reconstruction is solved as the inverse problem that aims to recover M from the known encoding operator and the acquired signal. In the case of undersampling, this equation is ill-posed, meaning that there are more unknowns than measurements. Therefore, a minimisation problem is used, where we can try to find the image which, when multiplied by the encoding matrix, results in the smallest error with the acquired data. However, this is still a difficult problem, and prior information is often incorporated into a regularisation term to constrain the solution. And there are lots of advanced reconstruction methods to recover good quality images, which differ in the types of prior knowledge which they use, including parallel imaging, which exploits spatial dependency of core sensitivities, temporal encoding, which exploits correlations through time, compressed sensing, which exploits sparsity in some known domain, and more recently, machine learning methods. But even if you are a maths wizard, how do we translate these complex methodologies into the clinical environment? As I've shown, these advanced imaging techniques often require the use of non-standard optimised sequences which are not available on all systems. The advanced reconstructions are mathematically complex, requiring gridding if they're non-Cartesian, and often have time-consuming iterative reconstructions. The data is often large in size, particularly when you're trying to perform high dimensionality imaging. Therefore, the reconstruction may require large amounts of compute power. And of course, this means that many of these advanced reconstructions are not readily available on clinical systems. And in the rest of this video, I'm going to talk about the different paths in which we consider when trying to translate advanced reconstructions into the clinical environment. So the first thing to note is that the raw MR data that we collect is in a different format depending on which scanner vendor you're using. This means that sharing of image reconstruction tools is not easily feasible, which hinders collaborations and reproducible research. However, this was recognised as a problem in the field back in 2013. By 2016, the ISMRM raw data standard had been designed by the MR community members. This common MR raw data format facilitates the sharing of data and reconstruction tools. The ISMRM-RD format is based on the HDF5 format, which packages together a combination of a flexible header section, which contain an arbitrary number of fields in an XML format, as well as a fixed structure raw data section that holds the data required from the experiment. Open source interfaces to translate MR data from the different vendors into this open source data format have been developed for C++, C, Python and MATLAB and are available from GitHub. The advantage of using this generic ISMRM-RD format is that we're able to use one of the many reconstruction toolboxes which are available regardless of the scanner vendor. And this slide shows some of the open source MR reconstruction toolboxes, each of which offers slightly different functionality or implementation in a different coding language. These toolboxes allow us to solve our own problems more rapidly by building on existing solutions. There are also a few websites which offer a more extensive and up-to-date overview of existing and new open source tools in magnetic resonance imaging. And these websites are a great place to become aware of the existing tools and compare their functionality to allow you to choose a toolbox which is most suited for your application. Of course, there are some other considerations which may influence the chosen reconstruction toolbox, which are influenced by how we're actually going to translate the novel reconstruction into the clinical environment. And there are two main ways of reconstructing the raw data: offline and online. These implementations have different advantages and disadvantages, and the pathway chosen may be dependent on the complexity of the reconstruction, the number of subjects it's to be used on, whether the images need to be viewed while the patient is on the table, and ultimately the skill set of the researchers involved. The chosen pathway will have an impact on the software languages, accessibility of other languages, and the hardware available. So what do I mean by an offline reconstruction? In an offline reconstruction, the data is not reconstructed at the scanner. Instead, someone comes and collects the data, often with a hard drive, and moves it onto another computer where they're able to run their reconstruction. The resulting images can then be taken to the clinician for analysis. There are many advantages to offline reconstructions, and this approach is heavily used in research, particularly in proof of principle studies. Offline reconstructions are really flexible, enabling any type of reconstruction algorithm, allowing implementation of the reconstruction in any chosen language, which opens the door to using any of the open source toolboxes and libraries. It also allows flexibility in terms of the hardware available, and this means that very computationally demanding reconstructions can be run on high-end GPUs, cluster computing, or even in the cloud. The disadvantages of offline reconstruction are, of course, that the images are not available at the scanner for visualization whilst the patient is on the table. The process of collecting the data and often manually starting the reconstruction and managing the resultant data is often performed manually. And the resultant images are not stored with the rest of the data for the patient. In fact, the resultant data is not often in a DICOM format at all, making it difficult to use standard post-processing tools. However, this recent study shows one example where offline reconstruction is used. In this study, continuously acquired 3D golden angle radial data was used to create motion-resolved 5D whole heart MRI images. Self-navigated cardiac and respiratory signals were calculated from the data itself, and the data was binned into 13 to 24 cardiac phases and 4 respiratory bins. The resultant 5D data was reconstructed using a compressed sensing algorithm, which exploits total variation sparsity along both the cardiac and respiratory dimensions. The reconstruction was performed offline on a high-end computer with a large amount of RAM memory and a Tesla GPU for high-precision parallel computing. This simple-to-use push-button framework enabled 5D whole heart MRI data to be collected in free breathing in just 4 to 8 minutes. However, the reconstruction time was about 7 hours per subject, which would make online reconstruction infeasible. The alternative to offline reconstruction is online reconstructions, where the images are reconstructed in a seamless manner at the scanner. There are a few approaches to online reconstructions. These include implementation of the reconstruction within the vendor environment, using a networked computer to perform the reconstruction where the data is automatically transferred from the vendor environment and back into the vendor environment, and of course ultimately getting the reconstruction implemented as a product by the vendor. I'll speak a little bit about each approach and the advantages and disadvantages of each. As we've already seen, each scanner vendor has a different data format, and so each vendor also has its own reconstruction environment. Despite their differences, each of the reconstruction pipelines is built up of multiple stages or processing blocks, which all have a different task. The data is passed from one block to the next to perform the complete reconstruction. Here is an example of a very simplified pipeline, where the data comes in on the left-hand side, goes through some blocks which perform different tasks, and ultimately the DICOM images are created. For some vendors, it may be possible to modify the existing pipeline. For example, it may be possible to remove a block and replace it, skip an entire block, or add a new block into the pipeline. There are some advantages of implementing your own reconstruction into the scanner reconstruction environment. The data is automatically reconstructed at the scanner as it is acquired, which means that the images can be visualised at the scanner. As you build upon existing functionality in the original pipeline, with careful coding, the data can be stored in DICOM format with all the other DICOM images with the correct header information. And this means the images can be used for planning of any subsequent scans and will be backed up with the rest of the study in the same way as all of the other scans. This approach is truly seamless from a user point of view, so no user input is necessary, and so it's very easy for the clinicians to use. The disadvantage of this approach is that there's often a steep learning curve in getting started writing your own online reconstructions. These environments normally use fixed coding languages with limited or no access to external libraries, and so the user often has to write new parts of the reconstruction from scratch. The other disadvantage is that the hardware which is available is fixed to whatever is on the vendor environment, where memory may be limited or GPUs may not be available. Of course, because the environment is vendor specific, it is not transferable between different vendors, and even between scanners of the same vendor you would need a research agreement to be able to share the reconstruction. However, this shows an example where online reconstruction has been performed within the scanner pipeline. The data here is a 3D stack of spiral sequence for time-resolved MR contrast, enhance angiography, where each volume is collected in approximately 1.3 seconds consecutively over 65 seconds. The modified online reconstruction includes an iterative non-Cartesian sense as well as a homodyne reconstruction. However, this is a time-consuming reconstruction which took approximately 1 hour to reconstruct all 50 volumes on the scanner hardware, which consisted of 2 CPUs. Of course, it might be possible to significantly reduce this reconstruction time with the use of more advanced parallel computing. And this is one of the potential advantages of moving towards online reconstructions on externally networked computing. There are a number of ways in which it's possible to perform reconstructions on external computers which communicate directly with the scanner environment, and some of which are shown here. The most commonly used open source vendor neutral framework is the Gadgetron. The Gadgetron framework can be integrated into the standard scanner reconstruction pipeline similarly to before. An emitter module and an injector module are inserted into the vendor reconstruction pipeline. The Gadgetron emitter is responsible for opening a connection, a TCP/IP, to the external computer and sending the parameters and the raw data, whereas the Gadgetron injector returns the reconstructed images to the scanner pipeline to achieve seamless integration within the clinical scanner. The Gadgetron reconstruction on the external computer consists of a series of modules or gadgets which are configured dynamically at runtime. There are many modules already available, but the user can also add new gadgets through a plugin-like architecture without recompiling the basic framework infrastructure. This functionality and flexibility makes the Gadgetron framework popular within the research community for online translation of advanced reconstructions. The advantages of network computing for online reconstructions include that data is automatically reconstructed at the scanner, allowing the images to be visualized at the scanner. Unlike reconstructions performed directly in the vendor environment, networked computing allows more flexibility in the use of programming languages, libraries, toolboxes which are available, as well as the hardware which can be used for reconstruction. The disadvantages of this approach, though, are that it is necessary to have an external computer networked to the scanner. There is a small additional overhead in terms of transmitting the data to and from the external computer, and the use of external computing and the reconstruction algorithms developed have not yet undergone regulatory approval. However, this shows an example where the Gadgetron has been used to perform low latency machine learning reconstruction for iterative real-time imaging. Undersampled radial data is collected from the scanner, which undergoes some processing in the vendor reconstruction pipeline, including coil compression. The Gadgetron emitter then transmits the data to an external computer where a series of functors perform a zero-field NUFFT reconstruction, which is then de-aliased using a previously trained ML model with convolutional long short-term memory blocks. And the de-aliased images are then retransmitted back to the vendor pipeline where they're available for visualization. As the application is interventional imaging, the overall latency for the reconstruction is important, and the paper showed that the measurement latency for the reconstruction was 39 milliseconds. However, note that 1/3 of this time is required for transmission of the data to and from the external computer. This Gadgetron-based ML reconstruction framework was tested in vivo for interactive imaging during right heart catheterization. Here you can see that the images are available on the scanner with low latency, allowing for interactive scan planning. All images appear in a seamless fashion, with the end user unaware that the reconstruction is happening on a networked computer. Despite the flexibility and impressive reconstructions which can be achieved with offline and custom-made online methods, unfortunately none of these methods are approved for clinical use. Therefore, the ultimate goal to truly get our advanced reconstructions to be translated into clinical setting, we have to get them turned into product by the vendors. This allows them to be disseminated to many sites where they can have maximum impact. To do this, the vendors have to undergo full regulatory approval, demonstrating accurate and reliable results. The disadvantage of using a product reconstruction is that the user has very limited control to make any adaptations. It can be challenging to get new reconstructions into product because of the amount of work which is required to undergo the regulatory medical device approval. Another disadvantage is that the product reconstructions only run on the hardware which is available in the scanner, and although more scanners are becoming equipped with GPUs, the hardware available is often fixed. However, there are some advanced reconstructions which have been FDA approved, including compressive sensing and more recently some machine learning. This has been adopted into vendor products and is available at many sites for use in the clinical environment. So in this talk, I've discussed the challenges and pathways for developing advanced reconstructions. Often these require the use of custom sequences which may not be available at all sites. I showed that there are many open source reconstruction toolboxes available which can help speed up development of new algorithms and improve collaboration and reproducibility. The use of the ISMRM raw data common format is important in enabling these generic reconstructions to be used regardless of which scanner vendor the data was acquired on. I've also showed different ways of using novel algorithms, including offline reconstructions and online reconstructions. However, ultimately, we should be aiming to get more of these advanced reconstruction methodologies through regulatory approval and into vendor product to enable wide-scale distribution, clinical translation and impact in patients. Thank you.